In this lesson we're looking at synapses and neurotransmitters in nerve signal transduction. So what we're looking at here is the bottom end or the axon terminal of one neuron sending a message across to its next neuron. Now you can see that they don't connect, and this whole join here is called the synapse, and that's where the signal is going to pass from one neuron to the next. But I want you to pay close attention that these don't actually physically touch, right? So the way that they send the signal is they turn what's considered an electrical signal across this gap or across the synapse to the next one, and that is going to be our nerve signal transduction. So a few important things here. When we talk about nerve impulses, what we're talking about is called an action potential. And what that means is it's an actual change in charge across the membrane. So we're talking a comparison between the outside of the cell membrane, remember our neurons are cells, from the outside of the cell to the inside of the cell. Okay, so we are often, just going to focus it here, what we see on the outside of the cell is different to what we see on the inside. So usually we have quite a lot of positive charges on this outside of the cell, and that makes this on the inside, even though they do have some positive charges, relatively negative. When this action potential passes down the axon, it actually reverses, right? Reverses the polarity. Because later on we're going to see that lots and lots of sodium and potassium ions flood into the cell, and that is going to switch, right? Okay, so let's talk about a few different things that have to happen before we get to there. Now, the very first thing that's going to happen as this message runs through the axon, we're going to be changing that polarity. What's going to happen, firstly, to kick all this off, is that some calcium is going to flood into the cell right at the back end of the axon, at that axon terminal. Now, because the calcium is flowing in, that's going to lead to what's called neurotransmitters start to be pushed in vesicles towards the end of the axon terminal, right? Now, by exocytosis, meaning that those vesicle bubbles are going to just connect and push out that chemical messenger or those neurotransmitters into the gap between one neuron and the next. Now that neurotransmitter molecule is going to cross the gap, and it's going to reach some receptors on the other side of the neuron. On the other neuron on the other side of the gap. Once those neuroreceptors bind, if they are matching up with the appropriate receptor, then it's going to lead to some more signal transduction, right? Once those neurotransmitters bind, it opens up ion channels, right? Ion channels open. And because they open, that is when we start to see the sodium flood into the cell. Right? The sodium, potassium will also do the same, but not in as bigger numbers. Right? It's mainly the sodium. Now, if enough of this sodium rushes into the cell, what happens is it reaches a threshold point, and that threshold point begins that depolarization, that change in polarity. Suddenly we are more positive on the inside and a relatively negative charge on the outside, and that's going to travel all the way down that neuron to the next part. And so if we had to reverse that and bring, okay, that's moving all the way down here, it's going to lead to that calcium come in, and at the end of that particular neuron, neurotransmitters will come out, push it, they'll move across the synapse, across to the receptors on the other neuron, and carry on. And that happens over and over and over again. Now, so many of these rush into the cell, that is what is causing that change in polarity, and that is what our action potential actually is. That's all our nerve signal transduction is. These neurotransmitters are those kinds of neurohormones we've seen and talked about once or twice. So these aren't necessarily hormones, but they are chemical messengers. And even though the movement along the neuron is considered to be an electrochemical charge there, this has to then translate to a chemical signal to cross the gap. because these two neurons are not touching. Right, now once that's all over, the neuron actually works to reverse this situation. So once the signal has travelled down the other end of the cell, in this direction of course, then the neuron actually says, well, we don't need all this stuff anymore, so we have to start to pump it out of the body. So we use active pumps to get rid of it. Now, we've talked about it before, but if we have the myelin sheath on the outside of this axon, that signal will travel much faster. All right, so with myelin about 200 meters per second it can travel, with no myelin about 0.5 meters per second, so not very fast. The pumps, as I said, work to get rid of all of this because all it is a change in charge. The action potential is simply different charges pushing inside the cell until it reaches a threshold and changes it completely. And that is literally what our nerve signal transduction is.